Hi viewers, I am Dr. Berlin Grace from Karunya University. I would like to explain the mechanism of DNA repair in molecular biology. Before going into the repair mechanism, let us see the overview about the types of mutation that happens in a cell. This can be categorized based on the cause or origin. It is spontaneous one or induced one. Induced mutation normally happens due to the tautomerism, depurination, deamination, slit strand mispairing, or replication slippage. This mutation also can be used by chemicals, physical, and biological agents, as mentioned in this slide. The effects of these mutations, whether it is a spontaneous or induced one, can be categorized into two types: small scale mutation effect and large scale mutation. Small scale mutation talks about the point mutation as well as multiple mutation. It can happen due to the substitution, deletion, addition, frame shift. And large scale mutation normally occurs due to the loss of heterogeneity, where the allele loss happens, and fusion is also one reason where we have translocation and inversion fusion. Deletion in bulk happens, or amplification or duplication of gene also happens. Let us see some major general errors that occur during the replication and natural repair system. DNA polymerase makes mistakes at a rate of 1 per every 1 lakh nucleotides, which means per cell division 1.2 lakh mistakes happen. Some of these are corrected immediately during the replication by the proofreading activity of the polymerase enzyme itself, or else it can be repaired immediately after the replication by the repair system. In both cases, if it is not happening, then it can accumulate lots of mutation. It can cause cancer. Some mismatch mutation can happen by deamination, as shown in the image. Cytosine, when deaminated, it is converted into uracil. So you can see the effect of such mutation in the DNA as illustrated. The U is introduced instead of cytosine. So when this DNA is replicated, there are two chances. One daughter will be having the same, it can have the same DNA, because in place of C, the C is inserted. Whereas in the other one, it can be converted into AU. Because of the U introduction, A can be added. So like this, deamination can cause mutation. Then tautomerism is another reason. Here we can see the example of the tautomeric A. It is converted into an immunoform. So this immunoform can base pair even with the C instead of T. Let us see the different types of repair mechanism that happens in prokaryotes. The estimated number of mismatch mutation that happens in E. coli is 1000 base pair. And the types of repair involves methylation mismatch repair, MMR, that fixes the replication error like base mismatches, or even small insertion deletions can be identified and that can be cleared. The mechanism involves a methylation in the old strand and unmethylation in the new strand, that we call it as a hemimethylation status. So the mechanism will be explained in the next slide. Then base excision repair, here it identifies the single base damage, and that base will be removed by glycosylase, and then polymerase and ligase will add new strand and fill the gap. Then nucleotide excision repair, a long stretch of nucleotide can be of an error one, and that can be removed by nucleotide excision repair called NER. It excises large DNA sequence and then the gap will be filled by ligase and DNA polymerase. The key differences when we look at MMR fits as replication error using methylation for the strand discrimination. BER fits a small base damage using specific glycosylase. It happens in a very short repair as patches. NER fits as bulky lesions. It recognizes helix distortion and it is causing a medium repair patches. Let us see the DNA methylation, the role of DNA methylation in the repair. And methylation is very important even for the prokaryotes and eukaryotes. They serve as a primary switch for gene activity. So let us explain the importance of DNA methylation. DNA methylation in prokaryotes happens in A in sequence GATC. So this occurs in the origin of replication as well as in the gene body. It can happen by DAM methylase enzyme after replication. In prokaryotes, the methylation is used for certain purposes. So all of you know that the restriction modification system is available in the prokaryotes, especially in E. coli. It is to protect it from its own restriction enzymes. So for this purpose also the methylation is useful. Then for the mutation repair system, methylation is essential. This methylation varies between the environmental conditions. Based on the environmental conditions, it can be of have a fluctuation to allow the rapid adaptation to the environmental stress or host immune responses. It will have an on-off methyl transferase system, or there will be a binding competition happening between the methyl transferase in methylation and the transcription proteins which carry out the transcription. So this will give a dynamic variation and act as a switch for the environmental adaptation. In eukaryotes also we have DNA methylation. It happens in the C, in CPG islands. This also is there throughout the gene. It can be there in the promoter region. And it is methylated by another enzyme called DNA methyltransferase 1 or DCM. It occurs largely after the replication to maintain the existing pattern. So the gene stability and the pattern will be followed for generation after generation by this methylation pattern. And here it acts as a long-term lock that stabilizes a cell's identity or silences harmful effect. The promoters have the CPG islands near the start site. This will regulate the gene expression. The unmethylated promoter will be giving an active transcription, whereas methylated promoter will silence the expression of gene. It blocks the transcription by physically blocking the activators, or it will recruit another protein called MBD, methyl CPG binding protein, which will recruit the histone deacetylase. When histone deacetylation happens, the gene expression will not happen. The DNA, the chromatin structure will be tightened into a heterochromatin form. Surprisingly, the CPG is also there in the gene body, which is methylated, but this will promote the active transcription. We can connect this concept with cancer, where you can see the tumor suppressor, it should be expressed normally due to the mutation or the epigenetic changes like this methylation. Lower methylation happens in the promoter. In that case, tumor suppressor's gene will not be expressed. Whereas another condition in the onco gene, due to the mutation or any other reason, this epigenetic changes happens where the hypermethylation happens. The methylation status will be, if it is reduced, then the transcription happens. The proto-oncogenes will be expressed in high level, that we call it as oncogenes. In prokaryotes, there is a crucial role for this DNA methylation in the repair that I am going to explain. As I mentioned, the A of GATC sequence in new strand get methylated only after 1 to 2 minutes post replication. So this period is very, very crucial. So the delay in this period also will facilitate such mismatch repair. During this 1 to 2 minutes, if any mistake happens during the replication, which is not corrected by the DNA polymerase enzyme, will be repaired here using repair system. The non-methylated new strand need to be distinguished for this MMR action. And the mechanism I'll be explaining in the next slide. It involves certain proteins and enzymes. Mainly mut-H will cut the cut the site where you have the mismatch, and exonuclease will remove the fragment including that mismatch region, and repair can be done by the other enzymatic systems. If both strands are methylated or none are methylated, such repair mechanism will not happen. So methylation plays vital role in MMR system in the prokaryotes. After repair only, the new strand will be methylated by DAM methylase in E. coli. So the hemimethylated state will be then there the CA protein will temporarily bind and act as a brake on DAM methylase. It prevents the premature methylation and facilitate or allow for the MMR to happen. That is described in this diagram. Mechanism of such MMR. Here the strand where the mismatch happens is recognized first by a complex called mut-S and L. It will thread through the DNA to form a loop. Then the strand discrimination is done based on the methylation. Mut-H that will discriminate the methylation pattern, and it is an endonuclease. It will make a cut by encountering the methylation status. It cleaves the unmethylated new strand at GATC sequence and mark it for repair. Then excision and resynthesis happens, and exonuclease will then one by one it will remove the nucleotide and make a longer gap. So that gap will be then filled by addition of nucleotide by polymerase enzyme, and ligase will fill the gap. It can happen from both the sides, from left side or right side, depending on the methyl site. Eukaryotic DNA mismatch repair, it is also called as MMR, but there is difference between the prokaryotic MMR and eukaryotic MMR. Prokaryotic MMR, as I mentioned, it is purely depending on the methylation pattern. Methylation is giving the mark. Whereas here Nick is giving the mark. Nick in the non-methylated new strand, that is mediating the MMR. So first the recognition is done by a complex called mut-S, and this will recognize single base mismatch or a nick or a small insertion or deletion loop. If it is a larger insertion deletion loop, that will be recognized by mut-S beta. So either alpha or beta, according to the lesion, according to the mismatch, it will bind, and then it will discriminate the strand based on the nick, and it will recruit PCNA. And this rely on the existing nick. The nick may be normally in the lagging strand we are having okazaki strands. Wherever we have the okazaki strands, there will be a nick, there will be a gap. So that can be repaired, or In the leading strand also, in the 3' end, we'll be having a primer initially that will be removed once the replication is over. So there again we'll be having a nick. So that can be also cleared here, or else a mismatch. So these lesions can be recognized, and the mut alpha or beta will bind. That will then recruit the PCNA. PCNA is now loaded into the DNA with the help of RFC1 protein, and this will guide the repair machinery. So now the repair machinery is assembled, the recruitment and insertion happens. Mut S alpha or beta, it recruit another complex called mut L alpha, which activate an intrinsic endonuclease which introduces additional nick. So now this nick will direct for the cleavage. So the excitation happens by another enzyme called exonuclease. So this will act from 5' to 3' and it will act from the nick site and remove the mismatch containing DNA segment. And the other, when the removal happens in the new strand, the template strand or old strand will be in single strand form. That will be stabilized by the RPA protein. Then the gap will be then sealed by addition of nucleotide with the help of DNA polymerase delta, and the gap is sealed by DNA ligase 1. So that is one type of repair called MMR. Now let us see the second type, that is base excision repair, where it happens as a patch. So here the base mismatch base is recognized, one base is not there or mismatch is there, that uh, mismatch base is removed by glycosylase enzyme that will scan and cleave the glycosidic bond so that the base is removed. But still it will retain the phosphodiester bond and the glycosidic linkage. So this will create an site called AP site, apurinic or apyrimidic site. And this site will invite an enzymatic system called phosphodiesterase. And this phosphodiesterase and one more enzyme AP endonuclease together, it will cut the DNA strand at AP site. It will cut the phosphodiester bond, and the sugar unit also is removed. This will be then repaired by addition of a new nucleotide, or a segment will be removed by DNA polymerase 1 and the gap will be filled by DNA polymerase 3 or 1. And the sealing is done by ligase enzyme 3 or 1, where is very common for prokaryotes as well as eukaryotes. There are two types in the burr. It can happen in a very small patch, or it can even happen as a long patch. Short patch is very common. It is the main mechanism where the polymerase beta remove the 3' PUA, unsaturated aldehyde PUA, that will be removed from the 3' end. And from the 5' end, the DRP will be removed. So the removal is done by the lyases or AP endonuclease. Then for long patch, in the 5' end, if it is resistant, then polymerase delta and epsilon, they synthesize 2 to 10 nucleotide. When it synthesizes 2 to 10 nucleotide, the existing strand will be then displaced as a flap. So that is removed by an endonuclease FEN1. So then the log gap created due to the removal of this flap will be then filled by ligase. In both cases, short patch, one nucleotide can be added and then that will be sealed. In the long patch, the new nucleotide added and that gap will be then finally filled by ligase 3. Ligase 3 fill the short patch, and ligase 1 fill the long patch. Sometimes the depurination also will cause the mutation. Such mutation will be also repaired by the same Baer mechanism. Here in the mismatch, the nucleotide will be removed and the AP site is created. But in the depurination case, already AP site is there because a purine is removed automatically or by spontaneous mutation. So then this gap is also filled by either by AP lyase, AP endonuclease action as a short patch method or as a long patch method. Long patch method occur only if there is a resistance for this action at the 5' end. Then nucleotide excision repair is third type of repair for mismatch. So here the mismatch normally happens because of the UV induced mutation. So the UV induced mutation, especially UVC and B rays, they induce covalent cyclobutane pyrimidine dimers, CPD. So this CPD, when it is there, then the new strand synthesis will be blocked. The replication will have a disturbance, and this will add by mistake. It will add a stretch of mismatch nucleotides there. So this can be removed by nucleotide. It will give a mutation as a long patch or long stretch. So this long nucleotide can be then excised out and it can be repaired. So this is done by a global genome NER GGR. So this detect and repair the DNA damage throughout the genome. It is initiated by a complex UVRA to UVRB. And the key protein involved include UVRA, B, C, and D, which is a helicase 2, then DNA polymerase 1. Then finally, the gap will be filled by ligase A. Mechanism is shown in this diagram. A dual incision happens and unwinding is done by UVRD because it is a helicase and it removes a patch of approximately 12 nucleotide, oligonucleotide, and then this medium patch will be then repaired. The same thing can happen during the transcription. So during the transcription also, if such mutation or cyclobutane ring is formed, then there the transcription will halt. RNA polymerase 2 will halt the transcription in eukaryotes, and then it will facilitate for such repair to happen. In human, if there is a deficiency in NER pathway, then it will lead to serious disease. Most notably, it is xeroderma pigmentosum, which causes extreme UV sensitivity. Then how this pyrimidine dimerization happens? It happens due to the UV light, especially C and B. This cyclobutane ring formation will not affect the base pairing, normal base pairing, but still it will disturb the replication process. It will disturb the transcription process. So this will introduce wrong nucleotide during replication in the opposite strand. And during transcription also it will disturb, so it will be halted and the repair system will act on it. And this dimerization can be repaired in the prokaryotes by different mechanism. First one is photoreactivation. Next, SOS repair and recombination repair. Photoreactivation is the direct, straightforward action where the problem occurs due to the UV light. Now that is rectified by light, normal light. So with the help of normal light and an enzyme called photolyase that will just reverse the reaction. It will cleave such covalent bond and reverse the situation. And this photolyase enzyme contains two cofactors like FADH and folate. It is light dependent process. However, this process will generate free radicals. This enzyme is present in many eukaryotes like yeast and plant, where it binds this CPD in light independent manner. But here the energy is coming from UVA or light, blue light. Blue light will give the energy to break the covalent bond. But it is absent in placental mammals. Hence, in mammals, if such type of mutation happens, that will be repaired by mostly NER, which I explained in the previous slide. 75 to 90% repair happens by NER. What is SOS repair? SOS repair is nothing but the trans dimer synthesis. It is error prone. So due to this, error can be there, but somehow the replication will proceed. Transdimer synthesis, it is survival is better than death. Replication system, it bypass or ignore and proceed across the dimer. So for that, the editing portion of this polymerase need to be silenced. For that, a protein called REC-A will bind, and this binding happens with the help of another two active facilitating protein called UMUC and UMUD. And these two proteins also will be synthesized, or the gene expression happens with the help of REC-A. REC-A will bind to the blocker LETS-A. LETS-A is the repressor that is cleaved by REC-A. It will help for the production of UMUC and D, and they will help for the binding of REC-A in the site of mutation. It bind there, and then it will bind to the epsilon subunit of the polymerase enzyme, and it will inhibit the process, it will halt the process, and it will suppress or silence the activity of epsilon of the polymerase, so that the polymerase will just skip this mutation area and proceed. Recombination repair, the same cyclobutane dimer formation is there, and the somehow with unknown mechanism the polymerase will proceed further, it will complete the replication. So there we will be getting a gap in the new strand. So that gap will be filled by a homologous DNA taken from the same cell, because during the immediately after the replication, before it is separated, we will be having two copies of DNA. So the correct sequence will be then exchanged from the it's a correct one, it is coming to the gap. Just an exchange happens, and the original place from where the right sequence is taken, we'll be getting a gap. So that gap will be then again filled by DNA polymerase 1. This is also error prone mechanism. And now single strand and double strand break is also there. It very often happens in the prokaryotic as well as eukaryotic system. And this can be cleared by the homologous recombination. Here it is not exchange. The homologous recombination repair happens by another method called invasion method. So the right sequence or the homologous sequence from another DNA or chromosome that will invade in this region, and the new strand will be synthesized. In eukaryotes also we have such repair system. More than 130 proteins are dedicated for such repair mechanism, and it is integrated with the DNA replication system itself. Loss of function of these proteins will result in genomic instability and an increased occurrence of oncogenesis. And just like SOS repair, we have translation synthesis. It will ignore the mutation and it will proceed. That is done by a special polymerase called ETA, DNA polymerase ETA. And several other low fidelity polymerases also will do this function, such as polymerase beta, then iota, and lambda, so on. Homologous recombination repair versus non-homologous recombination. These are two ways to fix the dangerous double-strand DNA breaks. Homologous recombination, it is a using a template from the undamaged correct chromosome or sister chromatin we can call, that will have long stretches of nearly identical sequence. So exchange of genetic materials happens by invasion of identical sequence, as I have explained in the previous slide. Accuracy, high fidelity is there. It's to restore the original DNA sequence. So it is error-free repair mechanism. It maintains the genetic stability and integrity. When it happens after the DNA replication, especially the S phase and then G2 phases it happens. The main role for this homologous recombination repair, it happens during the meiosis. Homologous exchange happens during the meiosis. Then in DNA repair, it happens for the double strand break, for accurate genome editing. In genetic engineering, this can be used for targeted genetic modification or manipulation to create the transgenic animals. In prokaryotes also it is useful for the process of transformation as well as conjugation. Non-homologous recombination happens. Here it is end joining. It is less common in prokaryotes. It is just like a quick fits, it happens in the eukaryotes. The complex called KU7080, it heterodimerize and then it will recognize the double strand DNA breaks and bind at broken ends, forming a ring-like structure. So that encircle the DNA and serve as a scaffold for the other proteins to come and bind. It recruit DNA PKCs. It is nothing but DNA dependent protein kinases. This will do the additional step if it is necessary. Like if the ends are not uniform, if we need to trim it and then make it compatible for the repair, all that process will be done by this complex DNA PKCs with the help of aromatase nuclease. So this complex together, it will function as an endonuclease and it will trim the incompatible or damaged ends. Then polymerase, micro or lambda can add the nucleotide to fill the gap. So these are all special polymerases. And then another complex called HRCC4 HLF ligase 4 complex. It joins the DNA ends and cause a permanent phosphodiester bond. Accuracy, if you look at it, is not accurate like homologous recombination. There, a correct sequence is changed by invasion method, whereas here it is done by a set of proteins. So some mismatch also happens, some mutation happens. It is very fast process, but it is highly mutagenic, error-prone method. Sometimes, if the homologous recombination is not possible or the template for exchange is not available, in that case, this non-homologous end joining method is essential. It becomes a crucial for survival of the cell. Thank you.